big hello to those currently at BioFest who will be watching this later.
Oh, that's right.
Yeah, that's where everyone is. So today I'm going to be talking about the epigenetic consequences of long-term exposure to antibiotics. And before we start, a bit of a caveat. When I say long-term, generally speaking, the average course of antibiotics is about 2 weeks. This experiment does look at some slightly longer time frames, but for reasons that we discussed later, it's still well within the realm of possibility for a good proportion of the human population to be exposed to these sorts of things. So it's not long-term in terms of 20 years of antibiotics from eating meat. This is from direct, it simulates intravenous exposure to antibiotics. So antibiotics, most commonly people are going to think of Louis Pasteur when they think of antibiotics. But he actually didn't invent antibiotics. He was one of the first to theorize about the antibiotic properties of various sorts of mold because he, among others, had noticed in the late 1800s that certain species of mold would inhibit the growth of microbes on, say, petri dishes in the lab. And the predominant theory at that time was that these mold species were creating a chemical that somehow killed off other living things, hence the name antibiotic. That being said, mold is not new. Since at least the time of the Romans, people had had bread. And since there's been bread, there's been moldy bread. People discovered pretty early on that very specific types of moldy bread could be eaten, and that would actually not have a adverse effect, but actually would cure you of other diseases. The other types of mold are, for example, I believe it was the Persian armies would leave special sorts of oil cakes to ferment by the hearth before they went on campaign for their wars. And then when they got wounded, they would slather this nasty, moldy, oily mixture all over their wounds, and it would prevent diseases from setting in. But actually, the antibiotics that I want to talk about most today are synthetic antibiotics, which the first of which was invented by these two well-dressed gentlemen. On the left, we have Paul Ehrlich, and on the right, we have Sadachiru Hata. In 1910, after a series of 605 failed experiments at creating a synthetic antibiotic, they made the first antibiotic compound that was not so antibiotic as to, kill the human body. So this has spawned a fair number of other synthetic antibiotics. But really, those didn't take off until Alexander Fleming found a reliable way to mass produce antibiotics, and they became actually fairly important to World War II, where they were widely used by the Allies, and subsequent use in the Korean War and other wars helped bring antibiotics to the mass market product we know today. As it stands, antibiotics have saved millions, if not tens of millions, or hundreds of millions of lives. And they are a fairly routine part of any sort of medical suite of treatments. They cure diseases that would otherwise be deadly, reducing them to, frankly, mere annoyances. And so the utility of antibiotics cannot be overstated. That being said, my apologies, I'm actually getting slightly ahead of myself. I want to talk about the various different kinds of antibiotics. So jumping back to those penicillins, this is sort of the difference between penicillins and cephalosporins. These are two of sort of the 8 broad sorts of antibiotics, and they're grouped in terms of how they function. So penicillins, as I mentioned earlier, are derived from various types of mold, and they inhibit cell wall synthesis, specifically in bacterial cell walls. They work on that final step of cell wall formation, preventing the cross-linking of peptidic glycan. As you can see, cephalosporins and penicillins are chemically almost identical. They're very, very close. Cephalosporins were invented to become less susceptible to beta-lactamases, which are produced by various bacteria to reduce their weaknesses to these molds' natural toxins. So both of these are incredibly widely used. Then another category of antibiotics is the fluoroquinolones. These actually enter the pathogen and inhibit the DNA synthesis. Specifically, they bind to the enzyme DNA complex within the nucleus of those cells. And again, there are a wide variety of these, all following this same basic structure. Then we have the sulfonamides. These are perhaps the smallest antibiotic. Generally, you have a fair number of other functional groups attached to this actual sulfonamide group, but these interfere with an early stage of folic acid synthesis, without which most living things cannot really survive. It's a structural analog of one of the compounds involved in that synthesis and acts as a competitive inhibitor. So you do actually need a fairly high concentration of sulfonamides to achieve that effect. These are known as sulfa drugs, and they were one of the first antibiotics to see really widespread usage in those years immediately following World War II. Then we have the macrolides, tetracyclines, and aminoglycosides. These all act on the ribosomes and the actual protein synthesis. Up top, these are all macrolides here. Then there's the tetracyclines and the aminoglycosides. Let's see, the macrolides specifically work on the peptidyltransferases involved in the ribosome synthesis of proteins. The tetracyclines irreversibly bind to the ribosomes and gum up the machinery that way. It is the aminoglycosides that bind irreversibly. The tetracyclines actually prevent the messenger RNA from binding to that ribosome, thus preventing protein synthesis and therefore stopping bacteria from, well, living. Finally, we have our nitrometazoles. These are going to be important later, as one of the compounds I'm going to be talking about is metronidazole. When these are administered, they actually exist as a prodrug when you give them to a human, because in their actual active form, they're toxic not only to bacteria, but also to all cells that we know of. However, anaerobic bacteria offer them such an environment to be reduced in such a way that you simply don't see them turning into that toxic compound inside your human cells. And so what's particularly interesting about these is that their toxicological properties are actually not cell-specific. That being said, each and every one of these has a number of side effects: nausea and vomiting, diarrhea, bloating, abdominal pain, but the NHS describes them as usually mild. In part, that's because these drugs have just been used for so long, it's hard to imagine a modern medical world without them. But as many people have pointed out, these antibiotics would likely not pass FDA approval were they introduced today because of some of the more severe side effects. One of the side effects that you may have heard of is growing antibiotic resistance. So there's Mrsa, which you may have heard of, methicillin-resistant staph infections. Those have developed, they have this gene, MEK-A, which encodes this protein which actually enables it to continue functioning in that cell wall synthesis, and it grants it a resistance to the beta-lactam family of drugs. That's your penicillins and related drugs. Antibiotic resistance has been greatly on the rise in the last few years, in part due to overprescription of the same antibiotics. So how it comes about is typically antibiotics are administered and they're effective at killing off 99 point however many nines you wish to add on of all bacteria. That being said, sometimes you'll get one or two who are lucky enough to have this mutation. They will then begin to spread, given that, oh, there are no other bacteria around. They don't have any competition for their food sources or for living space. And so they will flourish and then transmit to other individuals. So antibiotic resistance from Mrsa and other strains has been on the rise, and this is very difficult to treat because you'll get resistance to one drug, and then it'll get resistance to another drug, and so on and so forth. You'll eventually get to superbug status, where particular variants of these diseases will be resistant to almost all known forms of antibiotics. And traditionally the way to treat superbugs is to hit them with a combination of 2 or 3 different antibiotics that they are not resistant to. That being said, as superbugs become resistant to more and more antibiotics, that becomes harder and harder to introduce those antibiotics that they have not seen yet. So that's what makes this number of antibiotics improved descending line here especially worrisome, because as we're having fewer and fewer new antibiotics, due to it not being an especially lucrative form of research and development in the pharmaceutical industry, we have fewer and fewer options for taking out these superbug status bacteria. And so they pose more and more of a threat, especially to those in the system. Another side effect that has come to light in recent years has been deleterious effects on the human microbiome of antibiotics, because they are so good at killing things. When you take a pill, it travels into your gut, and your gut flora bear the brunt of that. So to take a step back, something which has, the importance of which has only become more and better realized in the last few years, is that within each of our large and small intestines, we have roughly 3 to 5 pounds of this gut flora, of sort of microbial roommates, you might put them. And we actually have co-evolved with this microbiota to the point where we rely on them to, train our immune system. Without this appropriate microbiota, prevailing theories are that this is one of the causes of allergies. We're almost certain that the lack of this microbiome causes autoimmune diseases such as Crohn's diseases through a lack of their ability to desensitize our immune system, to downregulate and say, no, it's okay, everything's fine. And it's also even been linked to obesity. And it's extraordinarily difficult to restore this gut microbiome, in part because some of it comes from the very first stages of your innate immune system response, in which you get some of your microbiome from your mother at birth. On the left here we have one example from a study which shows just how effective antibiotics are at killing bacteria. So on the bottom here, we've got a graph of the effects of various different types of antibiotics on the levels of C. diff, a fairly common diarrheal disease. So we see here that if you start and you don't take the antibiotics, you're obviously going to get a massive uptick in C. diff count. But with various types of antibiotic, you're knocking it down approximately 1,000-fold, bringing it to levels where your immune system can handle it, where it's not causing symptoms, and it's no longer an issue. That being said, when you take these drugs, they kill not only the C. diff, but they also kill off a variety of other helpful microbes that exist in your gut. And it affects not only the quantity of microbes, but also the diversity of microbes, killing off entire species. If you'll see how these pie charts are nice and happy and diverse, specifically vancomycin and metronidazole, if you'll remember those names, those will come up later. But those two are implicated to really reduce the diversity of your gut microbiome upon treatment. So then one of the side effects that has not been appropriately studied, however, are epigenetic changes, in part because epigenetics have, well, we simply didn't know about epigenetics up until a very few decades ago. So some epigenetic changes which are known to have effects on humans. If you'll all remember, it came to light within most of our lifetimes, I would say, actually. That bisphenol A, mostly in water bottles, is how I remember hearing about it, is bad for you in part because it's a xenoestrogen. It mimics estrogen, and part of what it does is it demethylates these CpG islands, which are specifically linked to an estrogen receptor promoter. And we're going to dive into the first half of this sentence here in about 5 slides. So if you'll just hold on to that for me, that would be lovely. Changes in these estrogen receptors and estrogen reception levels have been very strongly linked to a variety of cancers. And it's been shown that these effects persist not only through you, but also through multiple generations. But epigenetic effects aren't limited only to chemicals. Environmental changes can also trigger epigenetic changes. For example, there have been a lot of starvation studies done surrounding World War II and basically tracking epigenetic markers across generations in Europe. There have been a bunch of studies in Sweden, for example, where they're correlating grandparents starving due to blockades or wartime rationing with increased obesity or other disease states in their grandchildren. And it's believed that these epigenetic markers can and will persist even longer than that. It's just simply that we haven't had records in isolated enough populations to show that in humans as of yet. So you can think of epigenetic markers as sort of little tags that hang out on your DNA that inform how you then take the DNA and turn it into you. But in order to properly understand epigenetics, we really have to back up and talk about genetics. So, here on the left, we've got your traditional Watson-Crick double helix model, with your DNA base pairs all lined up. And on the right here, there's sort of a more chemical model. And the main takeaway from this is these are individual atoms here, so even very minor changes on your DNA is going to massively affect how they're going to be read by your cellular machinery. Epigenetic changes don't have to be terribly large in order to have these dramatic effects. So the other part of genetics that, the other image that genetics conjures up are chromosomes here on the right. Each of these chromosomes has, well, for us in this room, most of these chromosomes have roughly 3 billion base pairs of DNA. And it's these A's and T's, G's and C's, adenine, thymine, cytosine, and guanine. A's bind to T's, C's bind to G's, and no other combinations are allowed. In jumping straight from the double helix up to the chromosomal level, we're leaving out a lot of very important sort of mid-levels of organization. In fact, the chromosomes that you see here on the right, about half of them by weight are proteins other than DNA. So there's this sort of mid-level of regulation that has been ignored, and it's time to sort of take a closer look at that. So here we see that mid-level on the right with these histones that look like basketballs, and you see how this double helix DNA actually gets wrapped around them, and that wrapping basically makes it more or less accessible to your DNA machinery, both to read off your genetic code and produce the various chemicals that make you you, but also to bind it up and keep it stored where it's not in use so that you're not producing inappropriate chemicals. So there's a variety of different modifications that happen, and a lot of them focus on these histones here. So for a couple of them, you've got acetylation of lysine, methylation of serine, methylation of lysine, and phosphorylation of serine on these histones. All of these serve to sort of deactivate the histones and encourage DNA to separate from there and therefore become more accessible. So by inducing these sorts of epigenetic changes, we can actually upregulate certain genes and the transcription, and therefore making the proteins that those genes encode for. Other types include this ubiquitination of lysine. This signals for DNA damage, and it's basically a signal flag which says, hey, DNA repair machinery, get over here and fix this. We've got a problem. But the one that I really want to focus on today is methylation of cytosines. And so this is different in that this is one of those epigenetic changes that's directly on the DNA. And there's a specific sort of cytosine methylation that I want to talk about called CpG methylation. So CpG methylation is the creation of 5-methylcytosine, that methylated cytosine, at cytosines as followed by guanines. Typically you see these as what are called CpG islands. So those are long repeats of CpGs interspersed among other bases. And generally when you see those islands, those are telling the histones to wind up this piece of DNA and keep it very tightly bound. And that has the effect of shutting off genes. This is critically important in XY chromosomal silencing, and so it determines biological sex. But it also has implications for chromosomal stability, because when DNA is bound up by histones, it's relatively protected from damage. This is actually the best studied epigenetic marker, and I'm going to tell you why. So if you take 200 cytosines here, generally speaking, out of all of them, 3 of those 200, or 1.5%, are going to be methylated, which is a fairly low percentage. If you see, say, a 40% change, then you're going to see maybe an additional 1 or 1 fewer cytosine methylated out of 200. So it's going to be very hard to detect. If you take instead 200 CGs, the cytosine is next to guanines, then the number of those that are methylated in the typical human cell is something more like 70 to 80%. So if you have percentage changes in that, that's going to have vast implications for your histone unraveling, and therefore in the proteins that are being expressed out of your genetic code. So if you bring it back to that mid-level, you're likely going to see CpG islands wrapped around those histones. And as I said, that does have some implications for chromosomal stability when they're seen in islands, but also when you see rogue methylated cytosine, as you do here, that's going to be more worrisome. When your body produces methylated cytosine, it's going to tend to be in those islands, which are, of course, tightly bound. They're mothballed for a while. But when it's just out on its own, then DNA repair machinery can access it, and that's actually a bad thing. So here we have a cytosine on the left, and we have this 5-methyl, this methylated cytosine in the middle. A fairly common issue that DNA has is that various bases will become deaminated. So that's, as you see here, deamination is turning this into this. And when that happens to a normal, healthy cytosine, your DNA repair machinery will see it and say, oh, that's fine. I know exactly what happened here. You used to be a cytosine. Now you're not a cytosine. I'm just going to turn you right back into one. However, when it comes across a 5-methylcytosine that's been deaminated, then it's going to actually mistake that 5-methylcytosine for a thymine. So we've just introduced a way that you can turn what's supposed to be a normal cytosine that can get tagged or untagged with this methyl group into a thymine. That's a mutation right there. That's a point mutation. So especially if you have these free 5-methylcytosines, that's going to be worrisome in terms of mutation, which I probably don't need to tell you is a bad thing. And part of the issue is that if you're taking drugs, you would theorize that the effects of that methylation would not be any methylation that results from taking a drug would likely not be as ordered as your body's mechanisms for methylating or demethylating DNA. And therefore, you would be more likely to get these islands and therefore more mutations. Which brings us to the experimental question, which is, what effects might antibiotics have on human epigenetic markers? And specifically, are we going to see changes in CpG methylation as associated with the use of the antibiotic vancomycin over a typical period of treatment? So this experiment had 3 general steps. There was growing and treating cells. Then we collected the DNA through a DNase kit from QIAGEN. And then we tested for this 5-MC, for this 5-methylcytosine, using what's known as an ELISA, and I'll get into more details on that later. So for growing the cells, we chose HeLa cells, and we grew them in little dishes with what's essentially extra fortified cow blood, basically. But HeLa cells come from Henrietta Lacks, HeLa, who died in 1951 of an especially aggressive cervical cancer. And while she was dying, a doctor harvested cells from her tumors and proceeded to run experiments on them. And they were so successful that that doctor proceeded to freeze and ship copies of those cells around the world. Her permission was not asked, and in fact, her family did not find out until decades after her death that her cells had been used in this manner. They were awarded a settlement, but of course that doesn't make this theft of her literal body right. And many human cell experiments owe a great deal to this woman. So it's important to take a moment to remember who these cells come from and sort of the history of injustice that even the best intentioned science comes from. So these cells were fed on bovine serum albumin enriched media. That's a mixture of growth hormones that we put in this nutrient-rich sort of cow broth. And those send signals to these cells that everything's normal, just continue to grow and divide as you would were you in a human body, because understandably cells don't really like being taken out of the human body and put in a plastic dish. That's not a typical environment. They were grown for 6 weeks with antibiotics administered at regular periods. And it's at this point that I want to talk about treatment schedule. So typically, when you are administered antibiotics, doctors will prescribe a 2-week course. You will take the antibiotics and then you will wait for 5 or so days to see if the disease that caused you to take antibiotics is still there. If you're still showing symptoms, then typically the doctor will re-prescribe another 2 weeks, and they will repeat this process until you are declared disease-free. So it might be more logical to use a 2-week time course, but especially in children with ear infections, or in a lot of other cases, you're going to be doing these 2 weeks on, 5 days off, 2 weeks on, 5 days off cycles for perhaps months on end. So 6 weeks is perhaps not typical, but it's certainly not unusual by the same token. These cells were passaged roughly 14 times. Some of them grew slightly slower than others, and so I believe it was somewhere between 12 and 14 passages. And when you passage cells, that means that they have divided and proliferated so much, they've grown so many new cells, that you actually had to scrape them off of the plate and transfer a small portion into a new plate, and then give them no food. If you fail to do this, then they will wind up gobbling up all the food available and eventually drown in their own waste. Every single time that these cells were passaged, we re-administered antibiotic or or solvent, so that they would get that fresh dose of antibiotic on a pretty regular schedule. This works out to about 1 dose of antibiotic every 3 days. And the targeted goal was 2 micrograms per milliliter of antibiotic. This is sort of a first order approximate of what your cells might experience were you to get IV treatment of the antibiotics we studied. And it was suspended in DMSO, which is just a fairly common solvent. It's very friendly by biological standards, perhaps even too friendly. And the antibiotics we use are vancomycin and metronidazole, which you may remember from earlier. So metronidazole, the experiments on metronidazole were performed by Sam Yang, so thank you, Sam. And it's this little guy here. It was developed in France in 1959, originally used as an anti-parasite drug, which is unusual. That there aren't a great number of anti-parasite drugs out there, and it was actually surprising that it was shown to have an antibiotic effect. It's from that Nitrometazoles family, so it's going to act by inhibiting DNA synthesis, or excuse me, nucleic acid synthesis, and it's specifically toxic to bacteria because of that reducing environment. The only reason it's not toxic in our cells is that our cells don't provide that same sort of environment for it to be reduced in. And there's very little resistance among bacteria to metronidazole, so it's pretty commonly used for these sorts of antibiotic-resistant superbugs. And the other drug we chose was vancomycin, which is huge. It's discovered in 1959 from dirt in, I believe Botswana. And this is one of those macrocide drugs. So this is going to be this is going to be acting on cell wall synthesis. Again, this is very effective against Mrsa specifically, but also a lot of those other superbugs. And there's very low uptake into the body when taken orally. So vancomycin will almost always be taken intravenously. Both of these medications are on the WHO's essential medicines list, which means that they are considered critical parts of any medical toolbox, and also that they are considered to be safe enough for almost anyone to take. And so they've been sort of passed over on a lot of these studies because they've existed from a time before a lot of these concerns have come up. But part of the reason why we chose these two drugs are they're two incredibly different mechanisms of action. So metronidazole, on acting on nucleic acid synthesis, has to get all the way into the cell, whereas vancomycin stays on the outside, and part of it is through this lovely animation. So if you have your metronidazole here, and you've got your cellular membrane, it's going to pass right through those pores, whereas vancomycin is going to stay on the outside. It's just going to kind of bounce right off your cell wall. And it's for that reason that vancomycin in particular was thought to have very low potential to react with your cells. It was because of that very low biological availability. As generally speaking, most traditional thought has it that so long as it stays outside the cell, its effects are going to be relatively minimal, so long as it's not actively tearing down your cellular membrane. And we may see that to not be the case. So after our experiments with metronidazole wound up not showing anything conclusive, we moved on to vancomycin as sort of next down the list on those essential medications. But not to spoil the story, the way that we wound up measuring these things was through a technique called an ELISA. So that's an enzyme-linked immunosorbent assay. And specifically, we were looking at these 5-methylcytosines. And the way that an ELISA works is you take DNA from those cells and you stick it to the bottom of this plate. And then you have your primary antibody bind to those 5-methylcytosines, those methylated cytosines, and then you have a universal secondary antibody bind to that primary antibody. And those have special tags here, which will lead to color development in a very specific wavelength. And you can measure how much color is absorbed when you shine light on it. And so basically, the more color absorbed, the more 5-methylcytosine you can presume to have in your sample. Here's the raw data from my experiment. So on the left here, we have cells raised without vancomycin, and on the right, we have the cells raised with vancomycin. It does look as if both of them have some outliers. I included them for completeness' sake. But it does sort of, on an eyeball level, look as if you have a cluster here and a cluster here, and it looks like they might possibly be different. It looks like they have about the same variance. Normally, this data would be presented as averages and standard deviations. However, in order to do probability testing on that to determine whether or not we can say that it's almost certainly not due to random chance, we have to assume a normal distribution of our data. That is to say, this bell curve shape. If we have a non-normal error or uneven variance or really one of any number of problems, then we can't use this typical t-test for significance that most people would use. So when examining this data, the cells that were raised with vancomycin did show roughly a bell curve shape. This is approximately normal, and I did monkey around with this data to get it looking nice when you see it in the histogram. So these are just binned in various absorbance levels. However, when you look at the cells raised without vancomycin, it's really, you can't even eyeball this as a normal distribution. So we had to use a different sort of significance testing, and that was the Mann-Whitney U-test. So this is important to note because it doesn't require the assumption of a normal distribution. And as part of it, it's a less powerful test than the t-test. So if you're seeing your Mann-Whitney U-test imply that your results are likely not just due to random chance of two samples drawn from the same thing, then you are certain that your t-test would also tell you that. So to simplify, the Mann-Whitney U-test is very, very unlikely to give you any false positives. It's unlikely to tell you that there is a difference when there actually isn't a difference. When we ran the Mann-Whitney U-test, it implies that there actually is some sort of statistical difference between the cells raised with and without vancomycin in terms of the amount of 5-methylcytosine at these CpG sites. Actually, interestingly enough, it seems that the cells that are raised with vancomycin have a lower absorption level. So that means that there is a correlation between vancomycin and decreased levels of 5-methylcytosine. And that's not something to cheer about. It's not that more methylation bad, less methylation good. Really, it's more normal methylation good, abnormal methylation bad. And this is really shocking in a variety of ways. But there are also a couple of concerns that this study raises. One big caveat is that the sampling involved in this study is a little tricky. So biologically, this is a sample of 1, which would immediately raise red flags. And that's saying that basically there was just one dish of vancomycin positive cells, one dish of vancomycin negative cells, and I was just passaging each of those cell lines along so that if in any one of them I pipetted something wrong once, then that is naturally going to throw off the data. And that's certainly a weakness of this study. That being said, we can be a little bit more confident through the use of subsampling. Because each dish contains a population of HeLa cells. This was run with a number of subsamples, a number of replicates that were split off at the very tail end of the experiment. So we have a certain amount of confidence, but of course we would like to run this test a lot more. One of the major issues is that running ELISA is actually quite expensive. That and passaging large numbers of cells gets incredibly time-consuming very quickly. But really, the biggest question raised by this study is, what's the mechanism? Vancomycin stays on the outside of the cell, so it's obviously not directly getting into the cell and interfering with the signaling pathway that way. And there's no microbiome that it could be directly affecting. We're pretty sure that it doesn't directly act on anything on the outside of your cell. So the logical conclusion is that some sort of receptor on your cell surface is binding the vancomycin, and that's leading to some sort of signaling pathway. But that opens up a big old can of worms, and because there are a ton of signaling pathways. But of course, this also begs the question, what other antibiotics have been assumed to be safe simply because they are too large to be taken up into your cells? Which other antibiotics or any of a number of variety of other compounds are being bound by your receptors and leading to tangible differences in your cells? And I think that that's kind of disturbing food for thought. So with that, I would like to thank just a small portion of the many people who've helped me along the way. So first, thanks to you all for coming. And I would really like to thank Kimberly Dixon, who's unable to be here today due to a prior commitment, who has been my mentor and advisor for this project. I shot her an email last fall and said, hey, do you have anything cool going on? I don't have any lab classes this year, and I really want to do something good with my time. And she has taken innumerable hours out of her days to help me out, to teach me how to do all this, so I would really like to thank her a lot. And then Wayne Kruger and Joan Stam in the biology stockroom for making all these experiments possible. Professor David Hall and the Chem 680 class for listening to my presentation and helping me learn how to best present this information. Professor Paiseki for helping me out in the lab a couple times, and the Lawrence University Biology Department. So, thank you.
Questions? Can you go back to your data? Or was there a question? Oh, we'll go there. Sorry.
Okay, so my cell biology is not existent. So you're going to have to pardon it if I ask this using improper terms.
Yeah.
Vat pools are able to come around and encase something that they want to bring into a cell. Mm-hmm. So that way, if it's too large for the cell, membrane mm-hmm. to bring it inside. Is there any indication that that would not happen, or has there been any study being like, well, that's my question.
Yeah, certainly. So I would say that's on the one hand that is entirely possible, and this is just a very preliminary hypothesis, and it's a super general one to just say receptors. One of the many things that receptors do is actually trigger that phagocytosis. So it's not entirely accurate to say that there is absolutely no way for vancomycin to get taken into a cell. That being said, I would imagine that if it's being taken up in that manner, it's likely going to be broken down. And well, of course, its components, the components of vancomycin could then have an effect. And again, cell biology, not really a strength of mine either. I wouldn't expect that to be the case because I would think that would be more of a macrophage type activity, and this is cervical cancer cell as opposed to an immune cell activity. But that's certainly a possible mechanism that it could be acting through.
The data, when we were looking at the data, the non-vancomycin had 12,
Yes.
And then the vancomycin had 7. Why are there uneven number of spots?
Oh, so actually, if you take a closer look, these are 3 dots that are very close together. And these are 1, 2, 3, 4, 5, 6, 7, 8. So it's actually 12 of both. So
Okay.
it was 6 subsamples of each, each done in duplicate. So there should be 12 dots if you manage to split them out across each one.
Okay.
And the manual EU test results held constant whether or not I was using, whether or not I was averaging the duplicates, whether I was using each of the 12 samples, whether I was, there's a way to do it with a small sample size and with a large sample size, but it was consistently producing significant results either way.
My question. Let's thank our speaker then.